So hi everyone, my name is Kimberly Ebernez, and the title of my project, which I did for my thesis in biology to achieve honors in biology, is discovering new transcription factors that affect HIV-1 expression. So to begin, we pretty much, I think all of us know that HIV is currently incurable, and it's incurable due to a population of cells where HIV has latently infected it. And these latent cells, as we call them, are able to persist in spite of treatment. So that's why people have to stay on treatment lifelong. You can't ever be brought off it because these latent cells could push more HIV RNA back into your body. But normally, these cells are not producing any viral RNA. So we wanted to look at latency at the transcriptional level, which is when the DNA is made into RNA. And as you can see, the transcriptional control that we currently know about HIV is very complex. And all of these different proteins and structures of the DNA all work together in order to either produce more HIV or repress its expression in your body. So specifically, we wanted to focus on transcription factors because there hasn't been, we likely think that the whole array of transcription factors that could interact with HIV haven't yet been identified. And we thought that maybe if we could identify more and then validate them, then we could learn more about transcriptional control as a whole since they all interact. So we utilized a yeast hybrid screen, which basically allowed us to test if the transcription factor interacted with this piece of DNA called the LTR that they commonly bind to. And the results of that are here. Black squares indicate that there was a positive binding event, and then white squares indicate there was not. And then we also looked at if it was present in the cells that HIV typically infects, which are CD4 T cells and myeloid cells. Yellow indicates that there was a high level of expression of that factor in that cell type. So specifically, we wanted to focus on KLF3 and KLF2 because of the fact that we think that these could be latency promoting factors because they are very strongly active in cells where latency commonly happens. So we looked at, first we overexpressed the factor because we wanted to see when it was in abundance what happened. So when the factor is overexpressed, you actually see a decrease in HIV expression, which indicates that there might be some sort of repressive behavior here. And then further, we wanted to see the opposite. Okay, when we take this factor completely away, what happens to the level of HIV? So we used siRNA to do this, which is just a method of removing the factor. So here you can see we actually did see a decrease in the factor. And then when we decreased the factor, HIV expression went up, which is again consistent with maybe it having a repression on HIV expression. So finally, our conclusions, what we could draw from this is that based on previous literature and also our own experiments, we know that KLF2 and KLF3 are repressing HIV directly by binding, but also there's evidence that they might be involved in cell signaling pathways. There's also evidence that they might be recruiting these other factors, these other proteins, that could be creating a more tight DNA structure, which will also make it more repressive and harder to transcribe HIV. And then here we also know that it could be recruiting other factors that already are known, like BLIMP1, to play a role in HIV repression. So we were able to create a whole model just from validating a few transcription factors, and that'd be really useful also in the future for potentially developing more therapies that target certain pathways. Thank you.